career investigators. We're continuing to have presentations from an early career investigator from each of the research networks and the DPACC this year as well. So first we're going to hear from Dr. Grace Jacobs, who's with the DPACC. Then we're going to hear from Dr. Ilvana Jafic, who is from the Prescient Research Network. And then finally, we'll hear from Michelle Worthington, who is from the ProNet Research Network. So we'll turn it over to you, Grace. Thank you.
Hi, thank you. I'll share my screen. Okay. Hi, everyone. My name's Grace Jacobs, and I'm a postdoctoral research fellow at the Psychiatry Neuroimaging Lab and a part of DPACC. So today I'm going to tell you a bit about an approach and some preliminary data related to individual deviations in white matter associated with clinical high risk for psychosis. And this data is from the Shanghai At-Risk for Psychosis program. So schizophrenia is commonly characterized as a disorder of altered brain structure and connectivity. And specifically, white matter structure is often measured using diffusion-weighted imaging, which measures structure by examining diffusion of water across the brain. So a recently developed diffusion method called free water imaging, developed by Dr. Ofer Pasternak, one of the leads of DPACC, is a diffusion method that characterizes the freely diffusing extracellular water in the brain. And we're able to then identify a more specific fractional anisotropy measure that is focused on the diffusing water within the intracellular. And so free water has been associated with inflammation, edema, and atrophy, and it's been found to be elevated in first episode schizophrenia. Corrected fractional anisotropy, or FAT, has also been found to be reduced in schizophrenia, similar to more typical measures of FA. There have also been studies looking at free water and FAT in clinical high risk for psychosis, or CHR. And these have found altered FAT in CHR compared to healthy controls, as well as lower FAT and higher free water in CHR who later convert compared to those who don't convert. However, findings across these few studies have been mixed, likely due to differences in samples, multiple sites, CHR criteria, and methods. We also know that individuals with CHR represent a very heterogeneous group, and by looking at group-based analyses, we might be missing important deviations within an individual. So an alternative to this is to use normative modeling to look at individual deviations in white matter. So normative modeling is a framework to understand participant level differences in relation to a reference model, in this case, healthy controls. And so if you look at the figure at the bottom, we can use healthy controls, shown here in gray, to identify a normative range of a brain structure measure across age. We can then look at how CHR participants deviate from this normative range. This is an alternative to looking at group average differences because we can identify individual differences. We can also look across the brain structure regions to identify a number of deviations or how many deviations a participant has. There have been studies that have examined individual deviations in brain structure in schizophrenia, although currently there are no studies that have looked in CHR. These papers that have examined structural deviations in schizophrenia have generally found that deviations are widespread across regions, and they vary in which regions have deviations across participants. There's a lot of heterogeneity in location of deviations. They've also found that a number of participants overlap with the normative range in certain areas. There's also evidence to suggest that looking at only extreme deviations might not be an efficient diagnostic indicator. So for this work, we wanted to create age-weighted and sex-specific normative models of white matter, corrected FA or FAT, free water, as well as regular FA as a reference, to examine individual deviations in adolescents and young adults at CHR. So for this very preliminary work, we wanted to first look at, are deviations widespread across white matter tracks as they are in schizophrenia? And specifically, are there more positive deviations in free water, but more negative deviations in corrected FA? We also wanted to know if deviations were heterogeneous in their location across CHR participants. So for this work, we used the data from the Shanghai At-Risk for Psychosis program, or SHARP. So this is a large study of 126 participants with CHR, 26 of whom go on to later convert to psychosis, as well as 86 healthy controls, all between the ages of 14 and 25. All the data was collected at a single site on a single scanner in Shanghai, and advantageously, they're predominantly medication naive at baseline and represent a help-seeking CHR group that was collected on their first outpatient assessment and assessed using CHR criteria from the SIPs. So for the imaging methods, we use multi-cell diffusion data from a single 3T scanner. After quality control and processing, we applied an in-house free water pipeline to divide voxels into isotropic free water compartments and a tissue compartment modeled using a diffusion tensor. Within that tissue compartment, we were able to pull out measures of corrected FA, and then within the free water compartment, we were able to pull out levels of free water. And we did this across 46 white matter tracks. For normative modeling, we used an approach that our group recently applied and published on, where we examined age-weighted and sex-specific normative models of white matter. Specifically, we used a Nadara Watson or NW estimator for a locally weighted average with a Gaussian kernel as a weighted function. So this is a nonlinear, non-parametric approach that's able to take into account variance in healthy controls using a leave-one-out method. We calculated Z-scores for CHR and healthy controls, and a cutoff of 1.96 for the Z-scores was used to determine abnormal positive or negative deviations. And so what we found was indeed that patterns of deviations in white matter are highly individualized and widespread across tracks in CHR. So we find that the majority of CHR participants have at least one deviation, and that there are more deviations in corrected FA compared to positive deviations in free water. However, we also find that even though the majority of participants, CHR participants, have deviations, these are distributed across white matter tracks. So within a single track, no more than 50% of CHR participants had positive deviations for free water, and tracks had up to 11% of CHR with negative deviations for corrected FA. This is compared to healthy controls who had fewer deviations than CHR participants. And importantly, within a single track, no more than 5% of healthy controls showed deviations in any one track. This is a glimpse of the tracks with the greatest number of deviations, with the percentage of CHR participants with deviations in brackets. So in summary, using normative modeling is a promising approach that allows us to examine individual deviations in white matter measures at a participant level. What we find in our kind of preliminary data is that CHR participants do indeed have widespread and heterogeneous deviations in white matter corrected FA, as well as free water, similar to participants with schizophrenia. And so our next steps are to look at this data more closely and determine, as well as determine if a greater number of deviations or abnormalities across tracks in a single participant is associated with increased symptom severity, cognition, global functioning, and outcomes. The DPACC group, specifically Core 3, is also looking at using individual deviations to investigate prediction models for CHR outcomes. We also want to cluster participants and examine subgroups of individual deviations. And both of these approaches will optimally integrate the complementary measures of corrected FA as well as free water. So thank you so much for everyone who's making this work possible. And thank you for listening.
Thank you very much, Grace. Very exciting. We have time for a quick question if anyone would like to ask. Looks very promising. And given that this is one of the approaches that we'll be using in this larger study, we'll like to see how that gets incorporated or considered for our overall data analysis.